All right, so welcome back to the pre-lunch session or post-coffee session, however you want to take it. So we have delighted to have Professor Maitreyi Narasimha of Tata Institute of Fundamental Research to give the first of our two lectures on morphogenesis. So Maitreyi is again a trained medical doctor like Ramrai. So she got a medical degree from St. John's Medical College in Bangalore. But then she went on to do her PhD on genomic imprinting in the mouse at Cambridge, and then did two postdocs, essentially investigating how adhesion molecules contribute to Drosophila morphogenesis, one at Maria Leptin's lab at University of Cologne, and the second one at Nick Brown's lab at University of Cambridge, following which she joined the faculty of TIFR in 2006. So in her lab, she primarily investigates the molecules and mechanics underlying morphogenesis and their interactions. So without further ado, let me invite Maitreyi to give a talk.
Thank you very much. So can everybody hear me? Yeah. Okay. So I'd like to begin by thanking the organizers for inviting me to come and speak at this workshop. I have two slots, one today, one tomorrow, and I'm still kind of trying to work out how I'm going to divide it. I'll sort of make a beginning, and for most part tomorrow I'll tell you about the different lines of work that we do in the lab. For much of today I'll try and introduce the motivation for the kinds of things we're interested in asking. So the broadest question we're interested in is to ask, how do you build living structures? And you can have living structures across multiple scales, from protein complexes or large multi-protein assemblies all the way to an organism. And actually, there are some parts of our work where we look at how you build a multi-protein complex, and other parts of our work where we're looking at how you build or shape a tissue, or how do you make and position an organ. What I thought I would do in today's lecture is try and take an example and sort of motivate an introduction to different aspects of how we look at this problem. So by now, all of you know what morphogenesis means, and it's literally the creation of shape. And morphogenesis refers to processes by which cells, tissues, organs, and organisms acquire the shape or form that's critical for their function. And again, Shashi already spoke about form and the endless varieties of beautiful forms that Darwin alluded to in his famous book. And here are some of my favorite examples of form, mostly to focus on form that operates not at the scale of the organism as much as the scale of, let's say, a group of cells. And so let's say we look at the eye spot of the butterfly, or the feather of a peacock, or the stripes of the zebra, or this sort of polygonally packed pigmentation pattern on the giraffe. I think a question that we've been interested in asking is, can you begin to explain how these various sort of modular patterns come about? You might see the same kind of pattern across multiple organisms in many different contexts. So in a way, we've been fascinated by how you build these modular patterns. And one of the things that Darwin did himself say is that, most of these complex and beautiful patterns actually arise from a very simple beginning. And so it's really not so much about how you diversified, how you built diversity in a sort of evolutionary or organismal context, as much as how do you create the multitude of patterns that you see in living organisms. And so just to go back to a little bit of history, Joseph Needham subsequently became more famous for his studies on China, said long time ago that the central problem of biology is the form problem. And he said that the hierarchy of relations, from the molecular structure of carbon to the equilibrium of the species and the ecological whole, will perhaps be the leading idea of the future. And in a way, it also alludes to sort of multi-scale integration in the acquisition of patterns. Shashi already spoke about the evolution of form in evolutionary time scales, and I just want to use this slide to say that taxonomy relies on grouping organisms based on some morphological conservation, right? Worms, flatworms are flatworms, they're flat, and they're worms, and so on and so forth. And so form is important on an evolutionary time scale. The evolution of form is also interesting in terms of, you know, how did complex multicellular patterns arise from, I mean, when did you begin to see complex multicellular patterns starting from single-celled organisms? And so some of the work that we've not been part of, but it's very fascinating, looking at how much of the machinery for making multicellular animals with all these patterns was in place in some common unicellular ancestor. And it does turn out that much of it was actually in place long before the origin of animals. And many of you will have seen trees of this kind that also have, you know, ancestry or diversification marked in millions of years, right? So just, I mean, you'll hear, I'm sure, from Professor Bityan and Nanjundaya about the fascinating dictyostelium that lives very different lives from being unicellular to being a more social being. And here at the bottom is the choanoflagellate, which is effectively a single cell organism, but that can form multicellular organizations that look like rosettes, right? And I'll come back to rosettes tomorrow. The thing that we've been interested in is the development of form on a developmental time scale, and by that I mean during the time over which a single-celled embryo and zygote actually acquires the form of the organism it's going to become. And what you see here are the stages in the development of a human embryo, in which, starting from this fertilized egg that forms upon the fertilization of sperm and an ovum, you see changes that happen not so fast earlier on, but then very rapidly later on. And even if you just zoom in on the first 23 days of human development, you will see that what looks just like a little blip, maybe on an ultrasound, is actually a very complex complexly organized multicellular entity, which is characterized first by the division of the zygote into, by cleavage into an aggregate, initially loose, but then this aggregate becomes sort of more compacted through cell interactions. And a feature that accompanies this sort of compaction is the distinction of two different types of cells, red and yellow, as you will see here, but also the development of microcavities within this egg that then eventually enlarge, so that what we know as the blastocyst has red cells and yellow cells and a cavity in between. So formation of cavities, which become more numerous as development goes on, is also a sort of common modular theme, as are the formation of loose aggregates and their compaction and the formation of layers. So I've sort of defined 4 modules here. One is the formation of an aggregate, loose aggregate, a compact aggregate, formation of layers, the formation of cavities. And then of course it starts to get quite complex. But if you look at how a tadpole is formed, you'll see that some of these early stages actually don't look very different. They're all characterized by cleavage, aggregation, compaction, cavity formation, layers, etc. So a set of features that characterize development are an increase in cell numbers, an increase in the types of cells, and if you can watch this happen in real time, you'll also see that it's accompanied by an increase in the complexity of the dynamics of individual cells that make it up, that eventually lead to geometric complexity. Now, again, if you go back to Ernst Haeckel's work, one of the things that he noted was that there's a remark. So he postulated this idea of ontogeny recapitulates phylogeny, and what you see here, I mean, I'm not going to go into that just now, but what you see here are developmental stages of many different organisms: fish, salamander, chick, pig, dog, rabbit, humans, etc. And so you can begin to ask the question, is there one way in which development happens? Is there one solution, or are there many solutions? And recent thought on this are that there are stages in the development of organisms that are quite divergent, and stages that look remarkably similar, and this is what the concept of the developmental hourglass model is. And I'll come back to this a little bit tomorrow. So what's fascinated people for a long time is to understand how organisms acquire form. I've put a set of 4 M's here that people have thought about, and you'll see here, not because I'm at Mathematical Sciences Institute, the maths is on top, and then mechanics, materials, and molecules, right? So these are four things that people have been thinking about. The other thing that people have also been thinking about is how you sort of integrate information from molecules, which Shashi again spoke a lot about, to shape or change the form of cells, and how do cells cooperate to build tissues, how do tissues organize to build a body plan. So just a few concepts that a little bit in the order in which the chronology with which they were sort of discussed. So one one of our heroes, D.R.C. Thompson, actually postulated this idea of a mathematical basis for morphogenesis in the 1950s, and in his book on growth and form, he said that biological form can reflect physical and mathematical principles. And he also argued that a certain mathematical aspect of morphology is not just helpful but actually essential to the proper study and comprehension of growth and form. Turing, again, I'm sure this must have also been discussed, put forward the idea of the chemical basis of morphogenesis, in which he argued that complex patterns could be formed by relatively simple partial differential equations. And these are the reaction diffusion equations that we know of today that are capable of generating a whole host of patterns, some of which you might recognize as the ones that I showed you. For example, this sort of pattern here could be the skin pigmentation on the giraffe, this could be the spots on the leopard, and this could be on a tuna or something like that. And Professor Nanjundaya will, I'm sure, show you cyclic AMP waves in relation to dictyostelium that look a bit like this. There was still another idea. I mean, remember, all of these are roughly, happening the first half of the 1900s. The thermodynamic basis of morphogenesis, which was suggested by Malcolm Steinberg and Holtfreter, who argued that the way you build layers from loose aggregates, I spoke about aggregates and layers, the way you build layers in living structures is not very different from the sorts of physical principles that operate to ensure that oil and water do not mix. So I mean, in 1955, they demonstrated a similar unmixing when cells from different layers of an early embryo were combined. And at that point, they hypothesized that embryos and cells within them must rely on differential affinity to organize these layers during development. And Steinberg proposed that the forces organizing these cells into layers are closely analogous to those organizing molecules of immiscible fluids. And this was how the differential adhesion hypothesis was born. And what he was actually trying to say was that different degrees of surface adhesion between cells cause them to reorganize spontaneously to minimize their interfacial free energy, right? So in this scheme of things, the idea that morphogenesis also has a genetic basis and that genes direct pattern formation was a relatively new idea, right? And it was in 1980 that Yanni Nusslein-Volhard, Eric Wieshaus, and Ed Lewis, who actually famously identified this four-winged fly, right, that has two pairs of wings instead of one pair of the wing and halter that Shashi was talking about. It was this sort of trio of people who won the Nobel Prize in 1995 that actually first established that genes are determinants of spatial organization in developing embryos, right? And then a little more recently, Eric Wieshaus, who's one of the people I just showed you in this picture, actually made this sort of, I mean, sounds very intuitive now, but he said what I did not realize until late in my development as a scientist is that morphology and cell biology are actually the same scientific areas, or at least that the latter provides the molecular explanation for the former. So in a way, development went from looking at just genes as regulators of pattern formation or form to genes that regulate what cells do in animal tissues. And so I think we're still in this era of this cell biological basis of morphogenesis, and as developmental biologists, many of us are now also developmental and cell biologists who do basic research to understand how organisms build structures and to find out if there are fundamental laws, and if we can explain what genes, how genes actually help build, form, elaborate a structure or establish patterns. And the question that motivates most people who call themselves developmental biologists are how do you make complex and diversely patterned tissues from simple cells? But there are also another class of people, tissue engineers, who are interested in applied research, who want to be able to build tissues of a particular shape or function in the hope that they could produce a useful device or build a useful artificial structure by using the laws or principles that developmental biologists have uncovered, right? And I would say that in the last 25 years, I'd say that developmental and cell biologists are largely also working closely with physicists to ensure the sort of return of the physical biology of morphogenesis. This was in a way pioneered also in the 1960s at the scale of cells or groups of cells by Abercrombie, Trinkaus and Steinberg, who I already spoke to you about. But it has been, I think the resurgence of the physical biology of morphogenesis has been largely due to our ability to visualize, manipulate and measure not just what cells and molecules are doing, but also what sorts of forces cells exert on their neighborhood, what sort of forces they experience. And that combined with modeling with physicists has, I think, contributed to I would say the appearance of arrows not between molecules, which is I think a trait of geneticists and biochemists, but of arrows that indicate force vectors. And so I think the way I'd like to sort of organize this lecture and the next is to sort of think about blueprints, materials and motifs for morphogenesis. And what you see here is from a review in Nature Reviews Materials that looks at the properties of materials used in construction, but are actually biologically derived, right? So here you can see the living structures from which some of the materials we're talking about have been derived. You can see the sort of mechanical measurements that people typically perform on these architectural materials that include working out, for example, the Young's modulus of a material, both parallel when in this configuration as well as this configuration. And what you see here is a structure that's actually built from materials that have come from a fungal cell wall, right? So what I want to sort of do is to think about what are the blueprints for building living structures, what kinds of materials are living structures made of, and how do the properties of those materials influence what shapes or functions or dynamics they take, and whether there are some common motifs that recur in different contexts in the acquisition of form. Before I get there, I just want to tell you what a cell has. If you think about the various ministries inside a cell, I'd say these are them. Of course, the information, I mean, or the classical information in a cell comes from the genome, which is present both in the nucleus and in the mitochondria. Cells require energy to deform themselves, and this again comes through metabolic pathways that happen largely in the mitochondria. Their ability to deform relies on the properties of the membrane that surrounds the cell and the cytoskeleton, about which I'll say a little bit more later on. The cytoskeletal filaments, which are essentially polymers, in conjunction with motors and through linkages with the membrane, can exert forces, but they can also help transport material between locations inside the cell. There's a sort of factory that contributes to new material that's needed for the cell to grow, and this includes organelles like the endoplasmic reticulum and ribosomes. And then there's signal transduction as distinct from the genome, in which proteins sort of talk to each other in distinct ways to communicate, to transduce signals, amplify them, or suppress them in cases. Okay, so I'm going to start with the example of a single cell, because I want to convince you that before we think of animals, it's useful to think about single cells, and to show you that single cells actually exhibit complex dynamic morphologies. And what you'll see in this movie, if this plays, is a video from David Rogers from the 1950s, and what you will see here is a neutrophil, right? The kind that helps you fight infection. What you'll see, these little dots are bacteria, and what this neutrophil is doing is just chasing this bacteria in the hope of engulfing it and eating it, which it finally does at this point. And in so doing, you'll have seen that it changed its shape dynamically, sort of in the direction, I mean, in a way that its membrane protrudes in the direction of the bacterium, and then the membrane deforms again so that the bacterium is actually taken in, right? So what's happened to this single cell? Of course, it is a neutrophil. Not all cells in the body are capable of being such experts at engulfing debris or bacteria. So first thing it had to do was to become a neutrophil. Then it had to sort of change its shape, polarize its migratory axis, so there was a leading edge and a trailing edge, and then actually generate forces so that it could could move. So if you start to think about how this happens, obviously you'll have to endow a cell with capabilities that a neutrophil has, and so I'll call that committing a cell to the neutrophil fate. And I'm not going to speak about the exquisite genetic regulation that operates to make a neutrophil, right? And much of this is information contained in the genome, of which both the coding regions and the non-coding regions, as well as the higher order organization of the DNA in the nucleus, contribute to the making of a neutrophil. And just to keep up with the theme of this meeting, you'll see that that's all been worked out about how you go from a stem cell that could become one of many different types of cells to actually becoming a neutrophil. So essentially you sort of go along this path to become a neutrophil. But if you took a different path, you would become something else. And this is the idea of Waddington's landscape, where you have a pluripotent cell that has some options of the paths it can take. Some of these are reversible, some of them are not, and so on and so forth. But that's not what I'm going to be focusing on today. Instead, I'm going to ask, how do you actually move and shape a neutrophil so that it can migrate either towards a bacterium or to a wound edge, and then actually facilitate, in this particular case, the healing of this wound. So you see that this ring here is a wound, and what's happened here is that the wound has virtually closed by this time. What you'll see was responsible for that was the fact that these neutrophils, they're called hemocytes, but let's forget, not worry about the name. These neutrophils actually migrated towards the wound as soon or shortly after the wound happened. And what you'll see is that progressively multiple neutrophils migrate towards the wound. You can see now that they're all oriented in the same way towards the wound, meaning that they have these frilly membranes that are pointing towards the wound, and then they get closer and closer to the wound and help close it up. But the moment the wound has closed, they sort of change direction and start going in random directions. So you could ask, what is the signal that causes a neutrophil to actually appear at the wound site, right? And so a number of different kinds of signals could attract cells to move towards it. And I just want to use this slide here to say that that signal doesn't always have to be a chemical that has a gradient, although this is really the most common way of generating a signal, right? So you can have a chemo attractant that forms a gradient to which these single cells are attracted. But as I said, there are other sorts of sorts of signals that could attract this single cell, and there are a whole set of mechanisms that are named haptotaxis, durotaxis, topotaxis, and galvanotaxis to highlight the fact that material properties of the substrate on which these cells are moving could form a gradient. For example, you could have a material that either has a gradient of stiffness or just two discrete regions of high and low stiffness that could preferentially attract these neutrophils. You could have topotaxis where some structural feature could be what attracts these cells, and these structural features need not necessarily be graded, but they could be periodically spaced like that. You could have electric currents that are also attractive, and gradients could then, the signals could be made into steep gradients, even if the initial gradients were shallow, through other mechanisms that could involve receptors and enzymes that could convert a shallow gradient into a steep one, and so on. So if you look at what that cell does in response to a wound, actually you'll see that it follows a trajectory that's different, a little different from this drunken guy's random walk. Invariably, you will see that there is persistence, meaning that the cell moves for a substantial duration in a directed way, rather than just having a random trajectory. So how is that signal, wherever it might originate, sensed? And without getting into details, I'll just say that there are receptors that are present on the surface of these neutrophils that recognize the chemoattractant or help recognize differences in stiffness, or there are other sort of topological gradients or spacing features that are generated by the alignment of extracellular fibers. I'll come to these entities later, that allow the signal to be sensed. And then once the signal is sensed, something has to happen within the cell so that forces are generated that allow directional movement, and obviously that includes a whole host of other components that allow these cytoskeletal polymers to organize themselves differently at, let's say, the end of the cell that's closer to the source relative to the end of the cell that's farther away from the source. And so that bit is what we call transmitting the signal, and that typically involves signaling molecules that signal largely to organize the cytoskeletal polymers, or to bring motors in association with those polymers to generate force asymmetrically, differentially at the front end and the rear end, in a way that allows the protrusion of the leading edge. So with a little bit of introduction to what this actually involves. So it involves changes in the way, in the physical properties as well as the geometry of the membrane, and the membrane is just a self-assembling phospholipid bilayer with lipid and protein mosaicism, meaning you have islands of lipids of one kind, small nano domains of proteins of different kinds, and so on. And the composition, the protein-lipid composition, contributes to physical properties of the membrane, including rigidity and curvature. And the nanoscale organization, what we call mosaicism here, actually allows localized action, meaning that you can have a certain response just at a localized region of the membrane rather than uniformly around it, so that you can polarize the membrane. The membrane also contributes to interactions between adjacent cells and to interactions between the cell and the substrate. And through these interactions, you can generate tension at cellular interfaces or traction forces. The cytoskeleton consists of self-assembling polymers, and their organization, for example, branches or bundles, depends on the proteins they interact with, and their ability to generate force depends on polymerization against a barrier like the membrane or anchorage to the membrane, and they invariably work with motor proteins. And we typically think of two different classes of cytoskeletal polymers: actin, that typically works with myosin to generate largely tensile stresses, microtubules that work with other motor proteins, dynein, kinesin, and help resist compression. I'll sort of, just say that these mosaicism, I mean, all of us learned in school that the plasma membrane is fluid and mosaic. What that really means, and we've learned a lot about this in recent years since Singer and Nicholson, but what that really means is that you have these little small nano islands of proteins, for example, that can form and disassemble because proteins and lipids within the membrane can sort of move rotationally and laterally. And this sort of organization, as I said, allows you to localize signals. Then, I'm not going to go into the functions of the membrane, but from a physics perspective, membrane properties, including its phase behavior, viscosity, rigidity, and thickness, are actually determined by the types of lipids and the densities in the membrane. Lipids also contribute to the curvature of the membrane. And these lipid rafts are also those regions in the membrane where you can have localized signaling. The reason we're sort of a little obsessed with the plasma membrane is that at the scale of the cell, it's one key element that allows shape change. And I'll show you, you already saw the neutrophil change its shape through putting out these protrusions. Cell membranes are highly inelastic. And the maximum elastic stretching of a membrane is only 4%. And so any increase or decrease in membrane area when a cell changes shape or changes its volume are conventionally thought to be due to mechanisms other than simple elastic stretching. And these mechanisms include what we call intracellular trafficking, in which membrane is recycled through various routes. So what's on the membrane can get internalized, and then it can go back and add membrane. And this way, the reservoirs of you can generate membrane reservoirs, you can modulate the amount of membrane. Membranes can also be bent by, as I said, lipid composition, but also by proteins that have shapes that influence the shape of the membrane. And of course, membrane can also be bent by the cytoskeleton itself. So those are the ways in which the plasma membrane can be shaped. And the other part of the plasma membrane or another integral component of the plasma membrane are molecules that we call adhesion molecules that act as bridges between adjacent cells. So the brown molecule, these molecules here form molecular bridges with similar molecules on the other cell, and through their links to the cytoskeleton, they can exert forces. So adhesion is another important component of the membrane that can modulate the mechanical properties of the membrane somewhat locally. So by that I mean that, you know, the formation of these bonds can alter contractile forces generated by the cytoskeleton differentially at the point of contact compared to regions of the membrane that aren't in contact with another cell. Likewise, there are proteins that allow the proteins that are integrated into the membrane that link to the substrate, for example, the surface on which that neutrophil was moving, that can generate traction forces by virtue of the fact that the insides of those proteins also connect up to the actin cytoskeleton. So you can think of these linkages as being capable, depending on the type of adhesion you have, of generating either of making either slip bonds or catch bonds, and to be able to generate traction forces. So so broadly at the level of the membrane, those are the things that that we need to think about. Neutrophils, as I said, are single cells that are migrating. They're not sticking to another cell when they're moving. So we're focusing on single cells. And the cytoskeleton, just a brief introduction to the cytoskeleton, is that it's a collection of self-assembling polymers, whether you think of actin or microtubules or intermediate filaments. And their network organization depends on interacting proteins. And as I already said, their ability to generate force depends on polymerization, or for that matter, depolymerization against a barrier like the membrane, or by anchoring them to the membrane. And that combined with the action of motor proteins. And so polymerization of actin can generate protrusive forces. This is how bacteria move. Bacteria hijack the actin host actin machinery to propel themselves like comets inside cells, and you'll see that the kind of branching that facilitates that is what we call sort of dendritic branching that does that. Actin polymerization also generates protrusive forces against the membrane of this neutrophil to allow it to chase that bug. And depending on how it's organized with myosin, and here you will see that the orientation of the filaments, which are also polarized, influences whether you'll have contraction or expansion. And the way in which multiple filaments are cross-linked can also give you differences in the magnitude of force and whether that force is isotropic, anisotropic, etc. So these are the cytoskeletal or the actin cytoskeleton modules. And then you have switches that can turn on contractility or promote actin dynamics, or turn off these processes. So there are switches that regulate them. Microtubules are also polymers that work with a different class of motors, but unlike actin, they're stiff hollow rods and they can't be easily bent, and so this property allows them to this property allows them to help a cell resist compressive forces. And again, like actin, microtubule polymerization against a membrane can generate force. Depolymerization against a membrane can also generate forces. In one case, you're pushing forces. In the other case, pulling forces. So broadly, I'm just going to say that, again, like actin, the way they interact, the way the polymers are oriented, the way they interact with motors can allow you to either change the length of a microtubule bundle or cause it to bundle, and the two systems, actin and microtubules, can work with each other to sort of join hands and generate more complex patterns of force. The last thing I want to introduce is a set of proteins that are called polarity complexes that allow polarization even in response to a chemotactic gradient, as for example in the neutrophil, which develops a front rear axis. So there's symmetry breaking from something that was not polarized to something that becomes very, very polarized, as you will see from the polarized distribution of a whole host of proteins. That also helps generate the force asymmetry between the front edge and the trailing edge. Okay, so so a simple way to think about how you can change the shape of a cell is, for example, when it has to move, is that it involves a whole complex crosstalk between things on the membrane and things like the cytoskeleton and membrane trafficking inside. And here are some cartoons that just sort of show this complexity to the extent that you might begin to think even within a migrating cell you have subcellular gradients. Some of them are probably real gradients, some of them are not gradients that allow a cell to exhibit directional migration. Some of these are signals, some of these are polymers, some of these are adhesion turnover gradients, some of these are gradients of switches, right? So even within a cell, you can think of all of these gradients. And so the reason I sort of focused on this single cell was to start thinking about how a single cell can organize its shape and dynamics, and then perhaps move to the, in the next set of lectures, to go from just having a framework of how a cell can change shape to trying to understand how you can sculpt a group of cells to take on multiple shapes, and how you can move organs rather than single cells to their final positions in the animal. So perhaps I will stop here today. I'm supposed to speak for an hour, right? And then in the lectures tomorrow, I'm just going to focus on how you can sculpt a fly embryo into its shape by focusing on tissue scale processes that we in the lab have been interested in understanding. And I'll use two processes that we study in the lab to highlight the fact that there's a lot of um, um, even in cells that are similar with respect to the to the sort of genomes that they express, there's a lot of behavioral heterogeneity in these cells. This behavioral heterogeneity is essential to sculpt the tissue, and I'm going to show you how that heterogeneity is partly mechanical and partly molecular, so that you can see how one can build structures through diversifying cells molecularly and mechanically. And what I'll also do is to take you through how you can build motifs that keep recurring in the development of tissues across organisms. So to think of those as multicellular architectures that are deployed frequently during development. So I'm going to stop here.
Thanks, Maitri.
I'll also just say I'm a little hard of hearing, and also always a little worse after I've just taken a flight. So I please speak out loud on the
Okay,
mic.
I'll do my best. Can you hear me? Can you hear me?
Not very well. So I think it was sort of going in and out a little bit when I was hearing other people ask questions.
Hello, can you hear me now?
Yeah, I can.
Yeah, so I have a couple of questions. How does a typical size of microtubule compare with that of an actin filament?
Say again?
How does a typical size of microtubule compare to that of an actin filament? Are they?
Okay, so bacterium's of the order of a micron or so, and this neutrophil could be 20 microns in diameter.
What about the sizes of the microtubule, a
Size
microtubule,
is a microtubule.
and an actin filament? How?
Okay, so the diameter of a microtubule is 25 nanometers, right? Its length can vary. Some microtubules can be very long, some can be quite short. Oftentimes inside cells, we think you see microtubule bundles rather than single microtubules. So people who work with microtubules on a cover slip typically work with single microtubules that are of this diameter, 25 nanometers. But the microtubules, and I'll show you images, movies of microtubules inside cells, these we think in large part occur as bundles that could contain, we don't know how many microtubules, but at least 10 perhaps.
Okay, and the next question would be, is there a molecular motor that links microtubule to an actin filament?
Is there a?
Molecular motor that links an actin filament to a microtubule?
Yeah, there are, so broadly, the most common classes of motors that work with microtubules belong to the dynein and kinesin family. But there are also special classes of myosin motors that work on microtubules.
I see. And then myosin binds with actin filament, so that particular myosin can perhaps bind to
Right.
both.
So there are several classes of myosins. Most myosins work with actin. There are some that can work on microtubules.
I see. And can they also walk on actin, the one that can walk on microtubules?
Yeah, so I think, I think they can, and some can also cross-link actin
I see.
and microtubules.
Okay. One last question.
Yeah.
So the integrin molecules, can they form cell-cell adhesion, or do they only bind
Can
with
they form?
cell-cell adhesion? The
Cell
integrin,
cell?
yeah.
Can
Can it,
integrins form cell-cell adhesion?
Yeah.
Is that what you're asking?
right. or is it just that the integrin binds
So
with the...
that's a very good question. I'll say that they've been classically studied as proteins that help bind ligands in the substrate. But if you think of, I suppose the first example for cell adhesion dependent on integrins is actually in immunology, where this idea of an immunological synapse, where you have two cells that come close to each other. That interaction actually does require integrin, and its ligand, which is not in this particular case secreted out, but is also a transmembrane protein. So, but that function is what we call heterotypic adhesion. By that I mean you're interacting, you're bringing two cells together through interactions with another transmembrane protein. We have some reason in the fly to believe that integrins could mediate cell adhesion, but whether they're doing it homotypically by interacting with integrins on the neighboring cell, or whether they're also doing it heterotypically, is something we're still trying to work out.
Hi, can you hear me?
Yeah, I can.
Yeah, so my question is what decides in which direction the actin filaments would grow? I mean,
What decides in which direction?
yeah, because let's say coming back to the video that you showed previously of David, like by David Rogers, right? That neutrophil chasing a bacteria. So the bacteria is like changing directions, and it has to also follow the bacteria, right? And it has to change the shape of the its cellular structure so that it can follow it.
I will actually show you some real-time movies of cells migrating in my next talk, but I'll also discuss what sets a sort of bias in the direction of migration. What is the effect of a chemoattractant gradient, right? Does it help cluster receptors on the membrane locally? Does it create a local signaling hotspot because there is more ligand bound? Does that then locally trigger actin reorganization underneath that part of the membrane? And how do you sustain this? Is there a feedback between signaling at the membrane and the cytoskeleton? So I'll sort of talk about some of these things, but largely I think the simple answer to your question just now is that with a chemoattractant, ligand binding to receptor in that concentration gradient. So let's say, these are experiments where people typically will take a neutrophil and put a bead coated with a chemoattractant. So you have a sort of dot source of chemoattractant, and where you can actually follow both the migration of the cell as well as the organization of the cytoskeleton and the organization of proteins in the membrane. So the thing that this ligand binding to the membrane does is to cause receptor clustering. One of the consequences of receptor clustering, or two of the consequences of receptor clustering, is that this protein clustering can also deform the membrane. It can induce curvature, but internally there are more intracellular domains of the receptor locally that can signal to the cytoskeleton. So when we say actin is moving, actin is getting reorganized very locally underneath the plasma membrane. And so there are methodologies in microscopy now.
So you're saying the local signaling is coming from the receptors.
Say again?
The local signaling there is some local signaling that is coming from the receptor end, that is allowing the actin to move.
You
I mean,
what is the signal from the receptor?
yeah, that is localized. That's why the actins are like
Yeah. So
growing in that direction.
again, I spoke, for example, about cadherins and integrins. I'll show you what's common between the two of them, aside from whether they mediate cell substrate. But there's a sort of basic module about, you know, how they link to the cytoskeleton and how different ways in which they link to the cytoskeleton could have different effects on actin. So sometimes if you just want to make an exploratory finger, not one that allows the cell to translocate, but you just want it to explore its environment, right? Then you don't want to branch the actin, you want to bundle it so you have one finger sticking out rather than a frill. And so how the same chemoattractant could generate two different types of actin organization very close to each other is also something that people have sort of thought about a lot.
Okay. Thank you.
Hi.
I
So
recognize you.
yeah, nice to meet you again, Maitri. So I have a question that where you are saying that the lipids in the membrane are crucial because of the curvature that has to be there in the protruding ends and all. So does the reshuffling of these lipids happen actively, as in as the force is being applied? Let's say there is some local proteins being activated which are changing the rigidity of the membrane or changing the lipid composition locally, or is it a more passive process, as in like the membrane is responding to the surface tension and then lipids are sort of organizing to minimize
So
energy?
you're sort of asking what's the cause and what's the consequence.
Kind of.
You're
I mean,
asking
but they're
whether
simultaneous,
lipids-
so that causality, not from that side, but they can be simultaneous processes in the sense that the actin polymerization and membrane reorganization, lipid reorganization, can be happening at the same time.
Can actin organization influence lipid organization?
So are there some active processes that is changing the lipid composition locally, where the membrane is protruding?
I'm just trying to remember if people have looked at that. Yeah, so I think there's this guy called David Fletcher who's been working with these giant unilamellar vesicles into which, you know, you can sort of also create something like an artificial cell only in the context of plasma membrane and actin cytoskeleton, in which, you know, you could modulate the composition of the membrane. You could also sort of put in factors for the actin cytoskeleton, and I think the sorts of things he has looked at is what happens to the membrane curvature. I need to sort of scratch my head and
Thank
see
you.
if I have an answer to your question.
Any other questions? All right, if not, let's thank Maitri one more time. So we break for lunch in the usual place.